Okay. Well, today's talk will be done by Dr. Luciana Luque. She's our guest at the Institute for a couple of months, and she got her degree as licenciada in physics at the University Nacional de Catamarca with honors back in 2013, where she specialized in quantum gravity. After that, she moved to France, to Emergent, to continue her studies in a master's degree on black hole entropy, and up to 2014. And later on, she changed a little bit the subject of her work, still pure physics, and she got her PhD in la Universidad Nacional de La Plata with honors working on wetting transitions in three-state blue Emery-Griffiths models in 2019. This is, well, quite an abstract subject, interesting in statistical physics. We know a bit on that topic, but it's probably off the interest of the kind of things that most people are doing in this institute. So then she herself underwent phase transition and changed completely the subject and started to work on multi-agent models for cancer growth, tumor growth. She became a postdoctoral researcher funded by CONICET, which is the sort of the equivalent of CSIC in Argentina. And she's currently working at the Institute of Physics and Biology in the University Nacional de La Plata, under the supervision of Professor Manuel Carlevaro, with whom we are currently collaborating. She's here because we share a European Union project which is dedicated to exchange of research staff, and we will be sharing institute and office for approximately 4 months until she's back to La Plata. The topic of her research here is mostly focused on modeling cancer growth, and well, my role there is mostly technical assistance and community manager. So I'm trying to get connections with other people who are doing real work on tumor growth here in Madrid and in some other parts of the world. And well, the guy who shares surname with me is actually my nephew, who's a doctor and is also well, interested in these things. So Luciana, please.
Thank you so much. Well, thank you, Enrique, for the introduction, and thank you to the organizers for the nice invitation. I'm so happy I've been here today to tell you about my work. As Enrique mentioned, I'm a postdoctoral researcher, and today I'm going to tell you about my postdoc project in which I'm developing a computational model, a multi-agent based model, to study immunotherapies in tumor microenvironments. This is a collaboration work we are doing with my advisor in Argentina, Manuel Carlevaro, with Enrique Lomba here, he needs no introduction, and with Enrique Rodriguez Lomba. He's a dermatologist, so he's our clinical contact in all this matter. So first, before I start, I would like to show you a little where I'm from. So I'm from Argentina, but I'm working particularly in the city of La Plata, which is the capital of Buenos Aires. And let me take a minute to tell you that how amazing is this city. It was planned before it was built, so it has this weirdly square shape, and the streets are, well, like normal street, but also diagonals that cross the entire city. And every 6 blocks, it has a park, so it has a lot of nature between all the buildings. So if you have some chance of visiting Argentina after this pandemic madness, I would strongly recommend you to visit La Plata. It's so close to Buenos Aires, it's just an hour away, and it's a beautiful city. So as Enrique mentioned, I am doing my postdoc research in the Institute of Physics of Liquids and biological systems. And it doesn't only depend on the CONICET, which is the National Council for Scientific and Technological Research, but it also depends on the National University of La Plata, in which I also teach. So I kind of love these two institutions, and I wanted to show you here. So before I start, I would like to to mention that I'm not a biologist. I'm a physicist, as Enrique told you. So I got my degree in physics, I got my PhD in physics. So I apologize in advance for my limited knowledge on the subject. Okay, so what's the main goal of this project we are working on? Well, our main goal is to set up a virtual lab, yes, to study the effects of immunotherapies and maybe other therapies as well. Why not? We can combine different types of therapies in tumor microenvironments. So what's the behind the scenes of this virtual lab? What are the steps we need to take to set up this virtual lab? Well, first of all, we need to understand the disease we want to study. In our case, it will be like tumor growth. Then we need to develop a model, a computational model, that recreates tumor growth, and most important, its interaction to the microenvironment. So we can have like a more realistic approach about how this disease works and how it will react or how it will respond to different type of treatments. So now that we have the computational model successfully working, we have to feed it to real data, like experimental data. And there are two reasons to do that. First of all, we can use it to validate our model. And second, one of the most important parts is that we can generate distributions, yes, to all the possible parameter values and sample from that to create a group of virtual patients. You know, in real life, if you apply a therapy to a patient and it works properly, and you go and apply the same therapy to another patient, it may fail. So in order to model this heterogeneity between the patients, we create these distributions of the parameters to create our group of virtual patients. Well, once that we have our virtual patients, what we have to do is run a bunch of treatments on them and see how the treatment work, what performed best, and maybe we can use all of those results to finally help or contribute to the clinical trials. So, long story short, we have to understand the disease. Yes, here. We have to develop the model. We have to create the virtual patients. We have to run the in silico trials on them. And we can use these results to finally contribute to clinical trials. We are kind of far away from that point right now. So today I'm going to tell you what we are doing now, which is the model. We are developing the model. So my talk today will be about this computational model. And one important thing, I don't want to spend most of the talk going on the mathematical details. I know no one likes that. So, but I would like to give you like a taste of what's inside the model. So I'm going to show you a bunch of equations, but don't worry, it's nothing too deep. Okay, what are the key parts of our model? First of all, we know that diffusion is really, really important. Like, you know, growth substrates and metabolites. We also know that cell communicates each other chemically by diffusion. And most important, if we want to introduce a drug, it has to diffuse in to reach the cell. So we have to be able to solve not just 1 or 2 diffusion equations, but 5 or 10. So yes, this is one of the most important parts of our model. Once we have this microenvironment, we need to model the cells inside, because we know that most of cell behavior depends on this microenvironment. We also know that cells interact each other mechanically, like they push each other or they stick to each other. And one of the most important things about cells is heterogeneity. That means that each cell has its own unique state and parameters. So we already have the microenvironment, we have the cells wandering around, and yes, we can run a copy of the model, which is a nice experiment or a good sampling, but it is not really science yet, because this is a stochastic model. So we need to study across all the parameter space, and most of all, we need to study across all the stochasticity of the system. So we need to run many copies of the model to finally find the parameters that best match the observations. So we need to use high-performance computing techniques. In this case here with Enrique, we are working on this improvement of the model that I will tell you later. But yes, it will be a great improvement because it will allow us to get to time and scales that so far are out of our reach right now. So the talk is gonna be like in this way. I'm gonna tell you about those key parts, and while I'm talking about those parts, I'm gonna comment about how we optimize the model. So first, we have the microenvironment. We model the microenvironment as a continuous domain, but it is discretized as a Cartesian mesh to gain more computational efficiency, as I mentioned before. So for each voxel here, that is a voxel means a volumetric pixel, we store a vector of chemical substrates. That means that you can store any substrate that you want to diffuse in the microenvironment. So to model this chemical microenvironment, we model this as a vector of reaction, diffusion, differential equations for this vector here. So the first two terms are the diffusion term and the decay term of each chemical substrate, and the last two terms correspond to the bulk, source, and uptake term. We use different kind of methods to optimize this solver, like the operator splitting, the locally one-dimensional method, and the Thomas algorithm, which is also known as the tri-diagonal matrix algorithm. And I'm not going through these methods in this talk, but I will be really happy to answer any of your questions later if you want to know more. We can also set up, optionally, the so-called Dirichlet nodes. The Dirichlet nodes allow us to overwrite the value of the chemical substrate in any voxel within the computational domain. This is really useful to model blood vessels or just Dirichlet conditions in the outer edges of the simulational domain. So to validate our diffusion solver, we tested its conversions against a one-dimensional problem with an analytical solution and compared our results with those obtained by BioFBM, which is the parallelized diffusive transport solver of the PhysiCell project, which in 2019 won a prize from the PLOS Computational Biology journal for public impact. So they plot their solution over space for different time steps, and We model the same and we obtain the same. They also wanted to check the convergence of their model, and they got that their model is first order accurate in time and second order accurate in space. And we tested that as well with our model, and we obtained mostly the same. That our model is first order accurate in time and second order accurate in space. So we can conclude that our model is working properly. So moving on to the next key part of our model, we have the cell. Since we are working with this multi-agent based model and we use that object-oriented computing technique, we take the cell as a separate software object or agent that acts independently. So like most classes in software, it has attributes that are the internal state that that specific agent can manipulate on its own. And it also has methods, which are functions that act upon these attributes. So in the context of biology, the attributes would be like the position, the size, in which particular part of the cell cycle the cell is. And the methods would be the cellular processes, like going through cell cycling, or going through a death process, or growth and change its volume. And one of the most important features of our model is that our cells are of lattice or in continuum. That means that the cells are allowed to move freely in our space. It means that they are not like confined to any lattice or any spatial arrangement. And why is this important? Usually the on-lattice model, the cells cannot change their volume, and they are free to move only to previously stated positions, like in a chessboard. You know where you are going to move. In off-lattice model, the cell can go wherever she wants. So we don't have that limitation. And I'm not saying that the other models are wrong, of course not, but to our aims, this model, this kind of approximation will give us a more realistic approach about how the dynamics of the cells work. So one of the most important data structure of the cell in our model is the phenotype. And we regard the phenotype as the key thing because cell agents are really about cell behavior. And phenotype give us the chance to organize in a hierarchical way the different elements of this behavior. Because biologists are going to kill me right now, but in our model, there are really only a couple of things that cell needs to do, like cycling and get ready to divide, maybe die. It has a volume that has control. It has the size and a shape, so it has geometry. It sticks to other cells and push other cells, so it has mechanics. And it has to secrete substrates to the microenvironment and uptake substrate from it. So yes, these are all the key behaviors that each cell does, but just at different rates. So what we do here is that we organize those processes by behavior, and each one of those blocks has its own attributes and methods. For example, cell cycle. We take the cell cycle as a directed graph in which the circles here, the nodes, are the phase of the cycle, and the edges here are the transition rates. Two things about those transition rates. First, one of them must be marked as a division transition so that cell can proliferate. And the other thing is that those transitions can be constant or stochastic. For example, one model that we use in our model, one cycle, sorry, that we use in our model is the Ki-67, which uses a nuclear stain to this cell proliferation protein that mostly expresses when the cell is proliferative, so around mitosis. And we matched the transitions rates, to match we tuned the transitions rate to match the mammary epithelial cells. And one important thing about our model is that any cell cycle time rate can be tuned to match different types of tumor growth. Depending on what type of cells are we studying, their cycle takes more time or less time, or they grow in a different way. So we can adjust those transition rates to match any type of cellular growth. And the other important thing is that they can be modified in any time of the simulation on an individual cellular basis, so they can reflect the state of the cell or the influence of the microenvironment on it. To model the volume variation, we use just a couple of partial equations, like the differential equations, that allows the cells to grow or to shrink towards some target volume that can value depending on the cell state or the microenvironment influence on it. We also model different sub-volumes of the cell because, for example, when the cell becomes necrotic, it loses all its fluid volume to finally get calcified. So we need to differentiate different sub-volumes of the cells. Like, for example, here we have a nuclear and cytoplasmic volume. We have the solid and the fluid fraction volume of the cell as well. And we can approximate that volume to a sphere and get the size of the cell. So we can also model different sub-sizes of the cell. If we put these two processes together, we can see how the cell volume varies as it goes through different cell cycles over time. And we can also see here that the number of cells and the number of divisions grows exponentially. And that was quite expected because at this point, this is an ideal growth. The cell has nothing to stop its growth. It has an infinite growth. So it was kind of expected at this point. Mechanics. To model the cell mechanics, we use just classical mechanics, yes? And because we use the inertialess assumption, for which we can consider the acceleration of the system as 0, we can update the cell velocity, cell position by computing its current velocity, which is based on a balance of forces acting upon the cells. We model two main types of forces, an adhesion forces and a repulsive force, which means that the cells doesn't want to get deformed. So it's a resistance to deformation. Yes, when two cells are close enough to get deformed, well, the cells push each other not to get deformed. To compute those forces, we use here interaction potentials that are based mainly on the cell size and their distance to other cells. So once we have the forces, we can compute the velocity, and with the velocity, we can update the cell position using the Adams-Bashforth method for computational efficiency. And now that we are talking about computational efficiency, we also use the so-called linked cells. The main idea of linked cells is to reduce the area in which I search for cells interaction. So if we put these three processes together, like cycling, volume changing, and mechanics, we can finally see a tumor growth, an ideal tumor growth, right? The videos are the same, It's the same tumor, but this one in the right is looking for closer, so we can appreciate in a better way the mechanics and the volume variation because of the cell cycle. We can see how the cells push each other or stick each other and how their volume varies. And because this is because how I mentioned before, because the cells have no restriction on their growth, they will grow and grow and grow to finally form, as we can see here, a compact tissue. In particular, because of the mechanics, it will grow until it forms a compact sphere, as we can see here. So now, yes, we have to talk about the secretion, which is one of the most important parts of the phenotype because it links the cell with the microenvironment. So this is really important in this type of models because now the cell can have oxygen or secrete some chemical substrates as well. And to model that, we use the equation I showed you at the beginning, but we add the cell term for uptake and sources by the cell. So here we can see the heat maps of the microenvironment for oxygen and an immunostimulatory factor that the cells secrete. And we can see that as the tumor grows, it consumes oxygen from the microenvironment and it secretes this immunostimulatory factor. So now that the cell can interact with the microenvironment and has limited resources, it can die. So we model two types of death processes in our model, apoptosis and necrosis. And I said processes just on purpose because at cell scale, death is not like instantaneous event. It's a whole process. So we don't say the cell is death. We say that the cell is dying. In fact, it takes energy from the it consumes energy from the microenvironment to degrade. So we are not talking about death cell, but dying cells. So if we let the tumor I showed you before growth over time, we can see that at some point it starts developing a necrotic core. Well, I put this one here just to show you that finally it gets a perfect sphere. I wasn't lying at all. But well, if we halve that sphere, we can see that the tumor developed a necrotic core, as we can see in real life. And if we look closer, we can see that the cells have different sizes, right? Like here, or for example, here as well. And this is because when cells enter the necrotic process, the first thing it does is to swollen. It's swollen and swollen until it doubles its volume, and then it starts to lose all its fluid fraction volume. So it starts to shrink. At some point, it will lose all its fluid fraction volume and will get calcified. And that's why we have different sizes. That means that the cells are in different stages of the necrotic death process. So now I'm not gonna talk anymore about what's inside the model. Now I'm gonna show you what can we do with our model. As I've mentioned before, this is a work in progress. So the results are preliminary results, right? We are still working on them. So one of the most important things in cells, as I mentioned before, is heterogeneity. And this is an interesting area in cancer research because most of, well, I don't know if most, but some of the therapies fail because of this heterogeneity into the tumor. So one therapist kills some cells, but cannot kill the other ones. So to study this heterogeneity, we seeded a tumor, and we assign a random value of a mutant oncoprotein to a cell. And, we consider that our mutations, that the cell will become more proliferative. So we adjusted our model so that the cell cycle entry rate will scale not only to oxygen, but with this oncoprotein as well. So an increased value of oncoprotein is it makes the cell more proliferative. So we can see here time-based morphologies of the tumor along with the changing values of different cell types. And we can see that the primary tumor began with a normal distribution, but at the end of the day, the most mutated cells, which are here the red one and the orange one, outnumber the others. Another example that we have is angiogenesis. That when cell gets hypoxic, that means that it doesn't have oxygen enough to survive. It secretes some growth factor that induces the formation of new blood vessels, like here in those pictures, to keep the cell alive, to maintain the tumor nourished. So we model that as well. We have no blood vessel here, and we have a blood vessel here crossing the microenvironment in the middle. And in the first case, we can see that the tumor developed a necrotic core. But in the other one, we do not have a necrotic core. We just have a couple of necrotic cells. The tumor is bigger. The cells, the more mutated cells, proliferated more than the previous one. And this next result is one of the most important results to me. I got really excited when I saw it. So brace yourselves, because this is where the magic happens. Look how Look how the most mutated cells found their way into the blood vessel. This is amazing, because that really happens in real life, and that's the previous stage metastasis. And let me explain you why I'm so excited about it. Because we didn't program it. So this is an emergent behavior that emerges from the simple rules that our agents, so our cells, are following. So when I saw this, I was really, really happy. This is the magic about the agent-based model. Okay. Another result is that we know that each cancer is like a whole unique universe. Yes, they grow different, they behave different. So we can tune some parameters in our model to match different type of tumor growth. For example, here, we can bias the growing direction of the cells to grow to the size. Or here, we can add an interaction with a basement membrane, for example, and we can see that their behaviors are completely different. And we can match those behaviors. That's it. We can match those behaviors, those different growing, with different types of cancer. Like here, for example, in the first one, we have a rapidly growing melanoma. And in the one in the bottom, we have a superficial spreading malignant melanoma. I've mentioned that we are working with a dermatologist. So another tune that we can make is that we can change the rates of oxygen uptake of the cells. So we can vary different types of tumor growth. We can see here an example when we plot the volume of the cell over time for different values of oxygen uptake rates. So the first one would be like the ideal growth I showed you at the beginning. And then we start to increase this uptake. And we can see that as we increase the oxygen uptake, well, the cell takes all the oxygen in the microenvironment, so the proliferation decreases. And we can see that in the volume, that the volume decreases as we increase the oxygen uptake. We can quantify that by computing the specific growth rate as it is defined here. And we can see that as the oxygen uptake grows, it decreases. And we can literally see that and the tumors. The one I've showed you at the beginning with a low oxygen uptake rate has this sphere form, but if it starts to take more oxygen, if it needs more oxygen to survive, it will develop this weird shape, and we have a huge necrotic core. The other example is that we know that in tumor microenvironments, cancer cells are not the only type of cells. So we can model different types of cells. For example, we have here a healthy tissue. So we have healthy cells, which are the green see-through cells. And we have a tumor growing inside, which has also, it grows to the size and it interacts with the basement membrane. And we can see how it looks way out through the wounded part of the tissue. And it grows and grows and grows until everything is crazy here. If we turn that tissue, we can see that the basement membrane is full of cancer cells. That usually happens in cancer like melanoma. And the last example I'm going to show you is the immunotherapy. Immunotherapy is an immune attack over this heterogeneous tumor. And how does it work? The cancer cell secretes some immunostimulatory factor. So the immune cells will perform a bias migration towards this immunostimulatory factor to finally reach the cancer cell. When they reach the cancer cells, they stick to them and try to induce apoptosis. So one important thing is that an increased value of oncoprotein, as I mentioned before with heterogeneity, will increase the probability of dying because of immune cell attack. So we can see here, again, a time-dependent morphologies of the tumor with and without immunotherapy. And we can see here in the growth curves that after we inject the therapy, will take some time because it takes time to the immune cells to reach the tumor. But we can see how the tumor decreases its growth. So it's amazing as well. So, well, I'm to finish this. Where are we and where are we going? Well, we're still developing the model, but we are slowly migrating to the next step, so to fit it to real data. Anyways, the people who work with computational models know that it is a never-ending story. You always find something to improve. So this is not like we are going away from this step. We are always going to be around that. And I don't want to finish this without mentioning how important is to this type of projects to the multidisciplinary work. Because for us physicists, yes, we can develop the model, but we can get that far. So we need to interact with different professionals like biologists or doctors or whatever to have some realistic results, to make some contribution. So I would like to thank here, the perspectives first, sorry. Perspectives. First of all, even though we have a lot of methods that optimize our model, we are working now with Enrique here on translating our model to work in GPUs. GPUs are graphic processing units, so that will allow us to run in parallel many copies of the program, and it will allow us to get time and scales that are out of our reach right now. So it will be an amazing improvement. The next one is to calibrate our model with experimental data, basically or primarily from organoids. Organoids are an amazing thing. I didn't know that there existed until just a couple of months ago. They are three-dimensional tumors that recreate tumor microenvironments. So they offer the opportunity to study the cells' interaction in a highly scalable, longitudinal, three-dimensional, and transferable way. It's amazing. So there are different types of getting these organoids, but one of them, which was mind-blowing to me, is that they take the patient's cancer cells and they grow them in the lab to finally get a real tumor, like a 3D tumor. It's amazing. For me, that I'm a physicist and it's all numbers and equations and computers, to see a real tumor in a vase was amazing. So we are starting a collaboration here before going on with a group in the CENIO, which is a National Cancer Institute. I know the translation exactly. Here in Madrid, that they are developing these organoids. So yes, this will be another one of our perspectives. And the last one, like ultimate goal of our project, would be to perform the in silico trials to elucidate the best conditions to optimize existing therapies to specific targets, with the ultimate aim to, contribute to clinical trials. And as I was mentioned before, I wanted to thank my work team for being so supportive and to trust this project from the beginning. Manuel Carlevaro is my advisor in Argentina. Well, Enrique Lomba is my advisor here. Enrique Rodriguez Lomba is our dermatologist. He's our clinical contact. And Maria is the leader of the group at the CENIO. So I would like to thank them for trust in this project and for the hard work they're doing. I would like to thank as well CONICET for finance my postdoc. And I would like to thank also the RISE program for funding my stay here. And of course, I would like to thank all of you for being an amazing audience and for the people virtually as well. So if you have any question, I will be happy to address any of your concerns. Thank you.
Well, sorry for this technical little problem. Thank you very much, Luciana, for your nice talk.
Thank you.
I agree with you that multidisciplinary work is very important.
Indeed.
And I think your research is a perfect example of a biophysics research.
Yes,
which
in
is
fact.
something that is very interesting for all of us.
Thank you.
So thank you very much.
Thank you.
I think it's time for questions. Yes, Juan.
Yes, that's an amazing question. And I have to repeat the question, I think, because, yes. Well, he's basically asking how are we implement in vivo results in our model, right? Yes, we are already implementing some in vivo results for tuning, for example, the cells interaction. For example, they check the density of a cell population in our in a plate, in a in vitro dish. I don't know how it's say. I think it's dish. They compute how density it is and how deformed the cells are, and they can compute one constant for this interaction potential I've mentioned before. So we can have this, the interaction I've shown you in the tumor was a real cell interaction, like in vivo interaction. And yes, now that we have to calibrate our model with real data, what we have to do is to take a bunch of data, like many data, because of the heterogeneity of this disease. You know, cancer is completely different from one person to another. So we have to take a lot of data and generate distributions from that data to the parameters that we need to put in our model. So there are some parameters like cell volume, cell cycling rates. There are some specific parameters that we need to tune. So we have this group of data, we create this distribution, and we sample from that and create different types of virtual patients. So that would be the way, I don't know if that answers your question, but that would be the way that we are planning on. For example, with Maria now, she has this information about the organoid growing. So she's giving us that data, and we perform some statistical analysis on that, and we can tune the parameters of our model. And we can compare how our model function with her organoids. So it's quite amazing. Yes. Sorry? Yes, it's a good question. Well, our model is general, so we can tune the parameters to match any type of cancer growth. But since we are collaborating with Maria right now at the CENIO, she is working with mammary cell cancer and lung cancers. So we are trying to migrate. We were adjusting our parameters to melanoma, as I showed you before, but now since we have these results of breast cancer and lung cancers, we are trying to migrate our model to that. Yes, so that would be the type of cancers we are aiming right now. Yes. Well, I'm going to repeat the question. She's asking how long does it take to make one simulation. Depending on the size of the tumor, depending on the size of the microenvironment, but you can have 5-minute simulations to like 15-hour simulations, for example. Yes. Thank you. Yes. Yes, like if I can predict some behavior. Okay. So he's asking first if I can explain why. I'm going to try to go back here. No, I can't. So it's okay. No, it doesn't matter. Okay. Anyway, he's asking if I can explain. Let's go back. Yes, we are getting closer. here. So he's asking if I can explain this behavior that the most mutated cells go into the blood vessel, and if I think that our model can predict some behavior. Yes, I can explain what happened. After all the exciting about the result, I thought, why does that happen? And here's the thing. The cycle entry rates scale proportionally to oxygen and to this oncoprotein. So the closer the cell is to an oxygen source, the faster it will grow. So for those cells that, here for example, you can see that we have a group of cells here, but we do not have a blood vessel here. So since we have a blood vessel here, the cells in this border will grow faster, and they will proliferate in that direction because they have more oxygen. So that's the explanation why they tend to go to the blood vessel, because they proliferate faster, not only because of the oxygen, but because of the oncoprotein. So since they have this oncoprotein that makes them grow faster, and they have more oxygen, so they grow really, really fast. And for your second question, our model, I don't know if it has like a real predictive power. Like if we do not modulate, we cannot predict things. We know that this is happening because the cell proliferates with oxygen, but we can see emergent behavior that we didn't program. So like in here, we can see that the cells will go into the blood vessel, but we know why that happened. We wasn't expecting that, but we knew that was going to happen. I don't know if that answers your question. Yeah, great. You're welcome.
I have a question.
Of course.
If I have understood correctly, you are modeling a developing system.
Yes.
It develops in time. So my question is, if you go back in time, can your model somehow or you can infer parameters that are more sensitive at the very onset of the tumor growth to learn something about which factors have promoted this tumor growth?
Well, that's a great question. I haven't thought about it. So let me see if I understand you. You mean that from the tumor, we go back in time and see what happens to the parameters, like in the simulation?
In the simulation, I wonder whether you can somehow infer which parameters in your model are more sensitive in this very early stage of the
yes,
growth to know something about something environmentally that is influencing in these parameters, to know something, to know which factors in the end are responsible for the tumor growth.
You know, I haven't thought about it. Yes, of course.
I guess the point is not that you really can figure out why the tumor happened and what's the cause of the tumor, because these are factors that you already put into the model. But what the model can do is tell you what makes the tumor more aggressive, to grow more rapidly. So you can actually figure out, once you fit your model to real data, you can figure out why these tumors are more aggressive than others, and how they can respond if in the end you know how to implement some sort of therapy to one therapy and not to another. And you can figure out how the model will saturate in time, if in the end, after a certain time, you will be able to saturate the growth of the tumor before it's completely invasive. So this kind of thing you can model once you put all the parameters and you fit to real data. As to the origin of the tumor itself, well, that's pure molecular biology. That's far from the computational model.
More questions? I'm going to see if there is. There are many of positive comments.
Oh, thank you so much.
Talks, there are no questions. So if there is no more questions, I think we can finish by thank you.
Thank you so much.
And answer to everybody. So thank you very much.